All right, kids, here we go. We got a little pop from the 60s and 70s going on in the background. Hopefully it's not too distracting. The title of this song is Remember. So yeah, it's a good thing to remember your notes from stats class. Collecting sample data is the 1-3 theme, and this is basically looking at how do we collect appropriately our data. And we'll look at what's called design of the experiment, okay? Collecting that sample data. One of the more famous experiments that's ever been run was in 1954, polio and the Salk vaccine. Mainly because of the number of people that were run through this particular experiment. We had some 400,000 children run through it, and half of them were given a placebo, half of them were given the vaccine, and they were decided by a coin flip, which seems somewhat flippant, pun intended there. But anyways, of those 33 that got the vaccine ended up with polio, while 115 got polio without the vaccine. And so this has helped us eradicate, largely, I don't know if polio is even a thing anymore, but from the planet. So we nailed it. Well done. There's basically two ways to collect data. You can do an experiment, like the polio vaccine one, or you can do an observational study. The experimental is that you apply a treatment, and then you observe the results, like what happened, right? Individuals and subjects or the experimental units, like if it's like a machine or animals or whatever, right? Observational study, no, we don't treat. We only observe. And then you measure. So of the two, experimental is better, largely because we can control the variable. But on the flip side of that, there's ethics involved. Like, think about those kids that lost the coin toss and ended up getting polio because they were in the control group, right? So there's harm. And finally, you have to have willing subjects. In the past, that wasn't too much of a concern. You would there's all sorts of scary stories of doing experiments on groups of people that were not willing. They were just because they didn't have any power and they were part of that experiment. Horrible stories. Anyways, so let's look at ice cream versus drownings, right? Is there correlation between ice cream eaten and drownings? And most of us would say this is crazy. There's nothing to do one with the other. And yet, if we did an observational study and you collected data on sales of ice cream, let me turn the music down a little bit. Get a little loud there. Collecting data on sales of ice cream. And then you collected data on drownings, and you put that data back to back, so to speak, and you compared it. You would notice that, as ice cream sales go up, drownings go up. As ice cream sales go down, drownings go down as well. Well, most of us upon observation would say there's nothing there. This is called a lurking variable. It's hidden. The lurking variable in this case is summertime. And we eat more ice cream in the summertime, and yes, there's more drownings in the summertime because it's hot and we get in the water. So we've got to be careful with our variables. If you were going to do this as an experimental, like does ice cream really cause it, you would treat the subjects with either ice cream or cake, and then you would note the number of drownings that occurred with your subjects, and then that would give you either yes or no. There is nothing here. Okay, good design. Really important as we set up experiments that we are able to have these characteristics. Number one, that you can replicate. And that means, number one, that we have more than one subject okay? Literally, you want like 100 or 1,000 or more. And you should be able to repeat this many times and get similar results. If you can't replicate your design, yeah, then that's, or your experiment, then it's a bad, okay? Blinding. Subjects shouldn't know if they're being treated, right? Did they get the vaccine or not? They should know that. Some experiments are run as double blinds, in which case, not only does the person...
Attention students. Eli
Oh,
Newman,
dear
please
Lord.
report to the student office. Eli Newman, please
But
report
also
to
the
student
person
office.
that's giving the treatment doesn't know either, okay? So that would be the difference between a blind and a double blind. Randomization. Subjects are assigned randomly to get the treatment. And there are roughly 6 ways to do that. And I'm just going to go into that right now. I'm categorized them as 4, 1, and 1, meaning there's technically 4, but then we make a 5th, and then there's technically a 6th, but the 6th isn't really a thing. It better not be a thing. All right, simple random. This is can be a little bit tricky. It shouldn't be tricky because it says simple random. Every sample size has an equal chance of being chosen. Okay, very hard to do this well. There is also something known as just a random sampling versus a simple random sampling. I'm not going to get into that. I don't know that I'm even going to have that as a test question. It might be an extra credit, something, I don't know. We'll talk about that a little bit more in class as we get through the week. Systematic sampling. You start randomly, pick one individual, one bacteria cell randomly, and then you follow a rule and you do like every 3rd bacteria that you run into, okay? You test it. So there's you start randomly, but then there's the rule. And by the way, these are all random, defined as random, meaning they have random in there somewhere, okay? I'm going to skip the convenience. I'm going to skip to stratified. I would actually write the convenience one at the very end, by the way. The stratified is that they're layers. So what you do is you break your data up into, fancy word, strata, and then you select randomly from each. So for example, you might go by grade, okay? So you've got like freshmen, you've got sophomores, you've got juniors and seniors, okay? And then from each of those groups, you would pick, let's say, 20 or 30, and then survey them, okay? Cluster. Here we break our population into groups. So the fancy word is you partition in groups. Then, and you might need to do that more than once, yeah? But we're gonna do that until we get little clusters that are small enough, and then you're gonna pick those clusters. And in each cluster, you're gonna sample all the members in each cluster, right? So you're gonna do what we would call a census of each cluster. So you don't go from every cluster, you pick one, right? That would be more a stratified. But here you would go all the members in each cluster, but each cluster is a representation of the whole group, so to speak. Finally, multi-stage. Multi-stage uses a combination of all of the above. And here's an example, and it will show you some of the different types of sampling going on there. U.S. Census and the Bureau of Labor. They're doing a population survey specifically on job, employment, that kind of thing. So they want to look at all the U.S. Well, that's a huge population to look at, okay? So they don't do this very frequently. In 2007, they did it, and they first of all break them up into clusters. There's 2007 different clusters in the U.S., and they break this by kind of a region. They look at metropolitan, suburban, and then rural areas, and they break those into clusters. I don't know how specifically, but then each of those clusters is broken up into 870 strata, okay? From those, they in each of those 870 strata in each one, I don't think it's like by age. I think it's more of a geography thing. Yeah, I'm not too sure I understand this, but then they finally go to a numbering system, and they cluster each of the strata into 824 numbered houses, in essence, is what they come down to. And from that, they grab groups of 4, and these 4 are what they would call, I was going to go contiguous, but I don't know that you know that word. I'll say connected. So these are addresses which are next to each other. So that when somebody goes and polls, they can do 4 houses in a row. That makes it easier than to have like true random. And then one person is interviewed of each of the, and I think they end up having 60,000 total homes, okay, in groups of 4. All right? And from that, then we can gather data, information about the nation as a whole. All right, types of observational studies. So that was, again, these are randomizations of experimental studies, right? So this is an observational study now. Observational studies, where we just look, is you can look to the past, in which case we call that a retrospective, or you can do a cross-sectional, which is you look at a particular moment in time, and that could be now, it could be last week, it could be in the future, but you do it from a particular moment in time. And then there is what we would call a prospective. And that one is you set up, this is I call the procrastinators. You set it up for the future. And you look at a group of people as they move through time, or animals, or whatever, okay? So those are the 3 types of observational studies that you can do. We've got to be careful with confounding variables. These are variables like the ice cream in the drowning. We see an effect, okay? But you aren't sure of the factors. Like, why is that causing that effect? And this is particularly important as we look at things about gender and race, because in the past, many times, studies were conducted and it was largely done on a particular race or a particular gender, male, excuse me, and then that data is like pretended that it's like belongs to everybody, when in fact it's not. It belongs to a particular subgroup, and we can't put it across the full population, okay? Those results. Types of experimental design. So in the experimental design, we have what we would call a complete random, which is you randomly assign the treatment and the placebo. Then you have a random block, and this is where you would break into similar characteristics. So you make blocks, yeah? It's kind of like the layers. And you assign the treatment and the placebo in each. Finally, you can have what we would call a matched pair. And the matched pair is typically a before and an after. So you look at what happens to a person before and then what happens to a person after. Sometimes this is frequently done with twins as well, because they carry a lot of similar characteristics, having the same parents. And there is rigorous control. And rigorous control is extremely tough to do. But here you assign individuals to groups so that each individual is given, that the given treatment is similar in ways that are relevant to the experiment. So as you look at a particular person and you decide whether they should be part of the treatment, you look at a host of factors behind them that would tell you whether or not they're valid or not for your treatment. These are very beneficial. They're very useful types of designs, but it is tough. It takes a lot of work to set it up, okay? Errors, and this is how we'll end the unit. There are different types of errors that occur. One of them is called a sampling error, and these are between sample results and results from the true population. So your sample, for some reason, doesn't reflect the full population, whether it was too small or there was a particular type of characteristic that your sample had that the population didn't. That's what we'd call a sampling error. Then there's the non-sampling error, and these are the more quote-unquote interesting ones. There is the error due to human factors, right? Either, maybe it could be a simple computation error, that somebody just doesn't add things up right. It could be a reluctance error, in which case somebody is not getting the good data because they're not able, people aren't honest. And then finally, another one would be bad analysis. And that would be a case where somebody does a poor analysis of the population, right? And then therefore they've got errors with how they gather their data. All right? Non-random sampling error. This is an error due to not getting a random sample. And here this is like a convenient or a voluntary are the most, those are the most popular non-random sampling errors, okay? You're just too lazy in setting it up and you just said, hey, I'm gonna stand in this corner and just ask people. It's gonna be nice and random. Well, it's not random, you're just lazy. There's your homework assignment. Hopefully you can tick through those pretty fast. Just looked at chapter 2. It should be a fun chapter to dive into. Lots of math and pictures that we're going to be drawing and creating. All right, best of luck.